Yeah, so I work at the Willem Institute, working in theoretical biology. Like I said, we're completely remote, so I'm not used to physical rooms. Uh, so yeah, we recently did some work in biomedicine. So we did a new foundational model for medicine using these discrete cellular automata models. And I'm like a fan of the Levin Lab and the guys' research, so I'm really interested in hearing your thoughts. Yeah. And this is with Stephen Wolf. Yeah, so our goal here is to try to develop kind of a foundational theory of medicine. So Stephen says in the beginning of the post, as in practice today, medicine is almost always about particulars. This has gone wrong, this is how to fix it. But might it also be possible to talk about medicine in a more general, more abstract way, and perhaps to create a framework in which one can study its essential features without engaging in all of its details? So the goal, we're not trying to derive specific actionable medical conclusions. Rather, we are trying to model computationally and make precise the general phenomena of medicine. So our model organism is a cellular automata. You guys have all heard of a cellular automata? Anyone hasn't? Okay, so yeah, the idea is we have a bunch of rules, and they're supposed to be the genotype of the model, and then the pattern is the phenotype. Um, yeah, so, and then we think of the kind of fitness or objective of the organism is the lifetime or the number of steps until it reaches that all white state. Uh, and so once we have that model organism, we can kind of frame the concept of disease as making single cell perturbations to that pattern. So on the left here is the unperturbed, like quote unquote, healthy state. And then we make single cell perturbations and we observe the resulting behavior. So the pattern one right here is a case where the perturbation didn't make a really an effect on the underlying details or the overall objective of the quote unquote organism. But then the other cases are cases, so this one here, we make a single cell change and the underlying details of the pattern changes. But if we think of the objective, the lifetime is kind of stays the same. So it's still in a healthy state. So it's kind of like a trivial change. Um, and then the other ones are cases where the underlying details change and the objective or the fitness is also perturbed. And this last case is kind of like a kind of we can think about it as an analog for something like cancer, where you have a change in the pattern and then it kind of leads to unbounded growth, kind of living past the cutoff line. Yeah, so then the idea is to take, once we have kind of these disease state models, we can then make a second perturbation and try to direct the pattern back to its original lifetime. Um, so these are cases of different examples of treatments. And in these first three, the treatment is kind of unsuccessful in bringing the pattern back to its original lifetime. But then in the other cases, we have like instances where it does bring the pattern back to its kind of achieves its objective again. And interestingly, like quite often, the way in which it does its or achieves its goal is different when you give it the treatment, but it still like reaches a similar lifetime. Although in this case is kind of the exception where the details are kind of all put back in place, so to speak. Yeah. Okay, so now that we have kind of had this idea of a disease state organism, we can look at many more perturbations and kind of try to classify them into different kind of categories, like the way they do in medicine. And so traditionally in Western medicine, it's kind of imagined that you can make these discrete categories and things will fit nicely into them. But interestingly, if you look at our model, so you can kind of plot different perturbed states in feature space using machine learning algorithms. And what's nice is you get these kind of like blobs or like in between the categories. So it's kind of saying that it's kind of showing how even with this really simple program, you have enough complexity where things don't fit nicely into categories as kind of medicine traditionally like assumes. Yeah, so another nice thing that we can do in our model is we can look at kind of like a high level metric for the state of the system. So the orange lines here are showing the width at each step over time. And each different orange line is a different kind of perturbation. And these are all possible perturbations. And the blue one is the quote unquote healthy state. And so we can kind of ask from this like high level metric, is it possible to predict whether the system will terminate at a healthy state or not? And so and you can already see from this like plot that it's going to be hard to do that. And so what we do is we give the width history. It's kind of like the medical history of the system to a machine learning algorithm. So here's like an example of the history. And then it's supposed to output the lifetime or whether the organism is like healthy or not. And what's interesting is that the machine learning algorithm does very poorly because and so even with this simple program, it's kind of already the machine learning algorithm is struggling to compress it and find the pattern. So like on the x-axis is the prediction, and on the y-axis is the actual. And so you have like if it was doing it right, it would be like a complete diagonal. But like once you get past like predictions 80, it starts to really do poorly at predicting based on the width history. And by the way, if you guys have questions, you can figure out. Yeah.
What do you mean by the prediction? Or the prediction of the width, the prediction of the lifetime length?
Yeah.
What are you talking about?
Yeah, so the prediction of the lifetime. So the input to the machine learning algorithm is the width history. So like the width at every step up to a certain point, in this case, 25 steps. And then we ask it, or in the training data, it gives it examples of what that lifetime was, right? Yeah. Okay, so looking at more treatments. So on the top row, we can see these are just a random sample of treatments. And as you'd expect, most of the treatments don't work. And then on the bottom row is cases where the treatments do indeed bring the lifetime back to its exact original state. And what's interesting is in this first panel, you kind of have a treatment that's like happens very early on. And it brings the system back to like almost exactly the way it was, which is not a coincidence. And what we'll see later is the more kind of aggressive or early treatments, early intervention leads to better kind of treatment outcomes. Yeah.
Sorry. Oh, yeah, I was just going to ask, are these treatments like intelligently designed, or are they effectively just a second perturbation?
Yeah, so what's nice is the model's simple enough where we can just enumerate all possible treatments, at least for like a single disease.
Okay.
And when I say disease, I mean like a single perturbation. But yeah, I was actually about to talk about we'll do more sophisticated kind of treatment selection in a second. Yes.
So is there any like material definition of a healthy state versus an unhealthy state? Is it an attractor of some kind, or is it just an arbitrary definition?
Yeah. Well, when we found these cellular automata, we ran a adaptive kind of evolution algorithm that searches for patterns that reach a certain lifetime, despite certain perturbations. So we're kind of evolving it to be robust.
Cool. So these are kind of selected for robustness.
Yep. And yes, this one is kind of an attractor. Yes.
What do the colors represent on these graphs?
Yeah, so the colors, the cellular automata has 4 states in this case: white, red, blue, and yellow. So yeah, it's just a particular type of cellular automata. And each color represents a different state of the cell.
Okay.
Yeah. Yeah.
Is there any reason that the top right one was not considered successful, since we are seeing an elongation of time? I mean, it looks like it's going or of a lifetime, but it does look like it's kind of changing shape to go towards the left. But,
Yeah.
it has a longer lifetime than healthy state, right?
Right, yeah, so. There's a little kind of subjectivity in where we make the cutoff line. But it's kind of just like the fitness function that we use, we define the length to be, say 105 steps. And we kind of want the system to grow and stop for it, almost like, in a biological system, you want it to be a particular size. Okay. So we can look at the space of all possible treatments. So this is a disease perturbation. On the left is kind of the quote unquote healthy state of the system. And then we have an example disease perturbation. And then the colors here correspond to the effectiveness of the treatment. So in the lighter kind of orange represents if you were to perturb that cell, you would get a shorter lifetime. And then a darker kind of orange or a dark red means that the treatment kind of made the organism live too long. But what's interesting is even with, again, this kind of minimal model, you kind of get a lot of complex, like the mapping between the location of the treatment and the effectiveness is very complex. So if you're at a single cell and then you move one to the right, you're not going to necessarily get the same kind of effect. And this is just more examples of kind of mapping the location of the treatment to the effectiveness. And, you do see kind of like certain patterns, like certain kind of reducibility. But then there's also cases where it just looks kind of random as to which treatments are effective.
Can I interrupt just a quick second? Sorry,
Yeah.
coming from that. Can you go back a slide? So can you say can you define the color bar again just one more time? Sorry, I tried to grab onto it, but I didn't get it.
Yeah. So okay, so this bar is showing like if, for example, if it lives past this cutoff length, then it's going to be the color of the cell is going to be dark red. And so if it were to live all the way, if it were to completely die right away, it would be this like lighter yellow. And then as you go in between those, the color gets warmer. Does that make sense?
Yeah, so in terms of if you're comparing this to the outcome given no initial single cell perturbation. So red would be like a treatment that it doesn't necessarily recover the goal, but actually overgrows. So if I made like a dumb analogy, that's like a cancerous growth. Whereas the light colors are you give a treatment and it dies like immediately. So you've almost made it worse.
Yeah.
Okay, cool. What color then would correspond to the treatment creating a CA that best matches the original pre-perturbation?
Yeah, so in this case, it's like the original one, it lives like just before the cutoff line, so it'd be like an orange.
Okay, so is it safe then anytime you show these graphs, is that orange color what I should kind of look for in terms of like best possible treatments?
Well, this is the last treatment sensitivity.
Okay, perfect. Okay, then okay, cool.
Yeah.
All right, that helps. Thank
you. Okay, so we can, as you kind of asked about, we can look at more sophisticated ways to apply treatment. So this is a case where we're just kind of accumulating treatment perturbations and keeping the ones that lead to a lifetime that's closer to the original. And as you'd expect, it's kind of more effective than just doing some single cell perturbation. And as I kind of mentioned before, we can do the analog of more aggressive treatment. So in this case, it's also kind of an adaptive process where we accumulate treatment perturbations. But now we're insisting that each perturbation happens at one step below the previous one. So it's kind of like slowing down time and allowing you to be more intricate with how you interface with the system. And the outcomes for this algorithm are better, as you kind of put it. So we can even look at the kind of the analog of genetic variation. So within the unperturbed pattern, there's rules that don't ever get used. And so what happens is when you change a single cell, sometimes those rules that weren't getting used get used. So patterns that look the same with the unperturbed case can look different when you give them a quote unquote disease. So we can use that to kind of model what happens in biological systems where you have similar phenotype in the healthy state, but then when you enter into the disease state, you get kind of different behavior, even if the disease is exactly the same. So what this is showing here is cases where you have like different kind of inactive genes or quote unquote genes, and showing how they can lead to different phenotypes. So this is kind of all the population case of that. So we can look at they're all given the same disease here, and they have different kind of quote unquote genomes, or what we think of as the rules. And so what's interesting is you kind of have this like complex array of different phenotypes, even with just changing a few rules in the underlying pattern. And so kind of extending that idea, we can give all the kind of genetic variants the same disease and the same treatment, and look at the kind of like the analog of like a clinical trial. And so what we see is, again, like a different diverse array of complex phenotypes, kind of potentially similar to how if in the sample group you can observe one effect, and then if you go to the whole population, you don't see that same effect. Yeah, so kind of the conclusions that we draw, and that Stephen is making, is that medicine is hard because you're fighting against computational irreducibility. So the way that biological systems have adapted, they kind of put together using very complex mechanisms, and because of that, as kind of the quote unquote medical observer, you are kind of coming face to face with that irreducibility. And so because of that, we kind of need tools that are computationally sophisticated enough to deal with that. And yeah, so future work, we're looking to kind of like concretize this work and try to connect it with effects from noise. That's it. Thank you to Stephen Wolf, Mike Merson, Richard Starr, James Miles, and David Hammock, and thank you to you guys. Yeah.
Curious, have you looked at any relationship between the specific perturbation rules and treatment rules, at least the relationship between the ones that are able to like neutralize each other?
Yeah, I would say you can look at like a matrix of all the possible diseases and all the possible treatments. And it's interesting because there's kind of clumps in that matrix. So it's like certain like kind of with the maps that I was showing, there's certain areas where treatments are more effective. And what we find also is that, early quote unquote intervention also tends to be more effective. Does that answer your question?
Yeah, I guess I'm wondering if there's some sort of like mapping between like the time at which a specific perturbation happens, and then like some kind of function that describes at like X amount of time from now what is the perturbation needed to correct the system.
Yeah, I would say it's just like it's pretty complicated.
Yeah.
So
yeah.
The mapping is yeah. It's yeah.
So I'm an experimental biologist. Kind of what I do all day is find ways of testing things and collecting data sets. And so I guess the thing that would be most useful for me is knowing what would your like dream data set look like, right?
Yeah.
Like if you could wave a magic wand that had a data set that would allow you to connect the theory that you have here with, the clinic. Obviously, it will be hard to implement, but I guess what would that sort of look like?
Yeah, I mean, we've looked at like, for example, human single cell kind of data sets where they make a genetic perturbation and then kind of observe the phenotype with like image data. But what we're kind of after is a metric that kind of models the fitness of the organism from a very like high level, if that makes sense. So like, so like again, we're trying to do like a very abstract model, so not any specific organism. And so we're looking for a data set that kind of can give us really an accurate model of or metric of the fitness. What that looks like, I don't know. I come from the computer science side, and we're kind of expanding our biological effort at the institute. So that's kind of like in the cards or the goal. Yeah.
Cool. Thanks.
Yeah.
Have you ever found an intervention or a treatment that does nothing, the cellular automaton?
What do you mean by nothing? Like the original perturbation? Like
Yeah, I
you
mean,
make a
I
single
guess in other words, is there any way to perturb it that, or can the CA deal with it? Like, if you change something, can it?
Yeah, so
Okay.
actually, this particular pattern I was showing, in most cases, it does deal with it because we kind of evolved it to be robust and it is an attractor. So yeah, most single cell perturbations, it kind of maintains its lifetime. If you mean like deal with it in terms of keep the exact same cells or kind of like go right back to its original state within a few steps, it does happen too, but it's more rare.
Okay.
Yeah.
So it's very cool. Thank you. And you know, I think one thing about this is that it's actually a pretty good model of medicine today, which is pretty successful in many ways at extending life, but it doesn't necessarily give you the same life you had before the issue, right? And so the fact that you're mostly, if I understand, correct me if I'm wrong, but you're tracking persistence as opposed to a specific pattern. You guys would make when you try to reverse the perturbations, you don't have a way of getting back to the same pattern. And I think to me, that looks like the next big step is to have a quantification of like how close are we to the original pattern, and be able to get back to not just persistence, but actually the pattern as well.
Yeah, yeah, I think that's a really kind of good line of thinking. Like, I would say the fitness function that we chose, which is the lifetime, is kind of subjective, as you guys kind of noted. And so the reason why we chose that fitness function is because when you do an adaptive evolution, if you choose a function that's too specific, quite often we end up finding that you get stuck. So if we were to evolve, like to try to get a pattern that was a very specific sequence, then it wouldn't work. And so because of that, there's kind of a lot of like, you can change a lot and still achieve the kind of objective. But yeah, we're interested in seeing like the relationship between the fitness function and what can be evolved to, right?
Yeah, I mean, I'm not surprised because it's actually extremely difficult to, I mean, the whole structure of CAs is such that it's very difficult to tweak rules in a particular way to get a particular outcome, right? Like that's really hard. And so yeah, I mean, and biology is actually very good at that. So I think that gap between these kind of models and what the biology is doing is the kind of the big thing that we need to encourage.
Yeah,
That's people like Martin Card and others who are really talking about health as opposed to disease is I think that stuff is going in the right direction, is trying to understand how do we not just ensure persistence, but actually get back to the actual state that you had before, whatever.
Yeah.
That's really hard.
Yeah. Well, yeah, I mean, I know from your from the paper that you worked on with the differentiable cellular automata, I'm interested in like how does differentiability, affect the ability for the system to evolve to, a very specific state.
I mean, so we have a number of things that talk about this kind of stuff. We also have a from Peter Smiley's work of a cellular automaton model that's like that does some of these things. If I haven't sent it to you yet, I will. The biggest issue all of this faces is the inverse problem of getting you've got generative rules on the molecular biology side, and then you've got large system scale anatomical and physiological outcomes, and everything works fine in one direction. So in other words, you can make simulators that just kind of run forward and tell you how you get from one to the other. Like solving it backwards is computationally impractical. Nobody knows what genes or molecules to tweak to make desired changes.
Yeah.
There's a few cases where that works, but the general case that's not computable. So biology is doing something different. That's I think the future of this. I don't think it's solving that inverse problem.
Yeah.
Can I ask a question? So yeah, a very interesting talk. But just looking at your model, it seems to me like your model is more of a model of development rather than the sort of entire lifespan, right? If you think about it for biological systems, they're a bit like a parabola where it goes up and then it goes down with aging and ultimately death. I wonder, could you train models to sort of fit that parabola like behavior instead. And then you could look at things like lifetime extension and stuff like that. And also another thing is that you could, rather than looking at perturbations that breaks the system, you could also sort of take it from, for example, a patient variability angle, where you look at what perturbation can cause a disease state in a population of patients. That might also allow you to access these fast biobanks, perhaps.
Yeah, so with the first two cases, we've looked at both of those. I mean, it kind of results in similar behavior to what we see just trying to get a longer and longer lifetime. And yeah, like Stephen actually talked about, perturbations that extend lifetime. Yeah, and then what was the third point?
It's more like rather than looking at a single system with a set of perturbations, looking at a population of systems with a single perturbation. Like to for example, explain why you have environmental exposure of asbestos, for example, that causes cancer, but it only causes cancer in a certain subset of patients.
Yeah.
So sort of to use the similar kind of model to answer these kind of questions, I think would be interesting as well. Oh yeah, sorry, I might have missed that.
Yeah, that's it.
Cool.
So one thing that we're pretty bad at in biology is predicting what of the many injuries that happen are going to kind of propagate into becoming a big problem, right? This happens like if you walk around in sunlight, you get like 30,000 mutations per half hour or something like that, and you usually don't get cancer, but sometimes you do. And so I wonder if something that could be used for this is just a way to look for types of injuries that are going to propagate and kind of so in this case the cellular automata wouldn't be the whole state of the organism, but kind of the cancer or the disease, and just what types of things tend to lead to that kind of out of control, big problems. And then once there's an idea of that, then maybe finding ways to fix those types of things. Just because we don't have that type of systems level.
Yeah, I think, and you guys probably have a good intuition for this. It's just about like feedback loops, like rules that lead to themselves tend to be really bad for something like this. Yeah, and yeah.
Cool, thank you.
Okay.
Okay. Is that it? Okay, thank you.
Yeah, thank you guys.